talk to you about today is a project that started when I was finishing up as a grad student here in Toronto in Charlie Boone's lab, and then continued as I moved on to Princeton. I don't know if there's a... Yep. Okay. Anyway, so many of you know that Toronto is a major center for high-throughput genetics, in particular in yeast. In Charlie Boone's lab and Brenda Andrews' lab, a few years ago, the synthetic genetic array technology was developed. And it's a high-throughput genetic method for constructing and analyzing double mutants in yeast. And the way it works is pretty simple. You have a query strain that carries a mutation in a gene of interest, and you cross it to an array of mutant strains that carry mutations in all the other strains in the genome. And through a number of automated selection steps, you can induce mating, sporulation, and eventually selection of double mutants, which are then going to be available to you in an arrayed format. And then you can test these double mutants together with the corresponding singles for any phenotype of interest. So originally, the reason why this was developed is to study synthetic sickness or lethality, which is a very simple type of interaction between genes, such that the cell can tolerate the loss of either gene alone, but not both. And this, in general, corresponds to cases where the two genes buffer for each other and impinge on the same essential biological function. Originally, this is how a typical SGA play looks like. You have 384 mutants present in quadruplicate, the 4 colonies right next to each other, and then you're looking for cases where the wild type and the 2 single mutants are perfectly viable, but the double mutant is not, and that's the case you're interested in. Of course, since the original studies, we took it further, and we can now measure quantitative phenotypes by measuring the area of the colony size from digital images of the plates. So we take digital images, we process the images and measure the colony areas, and that's our quantitative fitness. Then we can then feed into a formula that calculates a genetic interaction score that determines how sick or how healthy your double mutant is compared to your expectation. And of course, this has been done on many thousands of gene pairs. In reality, the goal is to do it all 6,000 by 6,000 double mutants in yeast. And in the first round of experiments and analysis, we've generated a bunch of maps that not only show you that genetic interactions can help you to cluster genes into functional modules, but also allows you to study connections between modules. But this is not what I wanted to talk about today. Today, I wanted to tell a short story about paying attention to little things. It's about the take-home message of this story is that SGA, just like any other genomic technology, has many layers of information hidden, and we should be paying attention and spending time thinking about what other information we can get from data we already have. Because to this day, this is a pretty expensive experiment to do, and we should make sure we squeeze out as much information as possible. So to tell you this story, I have to remind you a couple of things about how SGA works and how double mutants are constructed. So this is basically a genetics 101. If you have 2 single mutants that are carrying mutations on 2 different chromosomes, then when you mate them and obtain the heterozygous double diploid, and you enforce sporulation, 25% of your meiotic progeny will carry both mutations. So that's under the assumption that the 2 chromosomes will segregate independently, which is usually a pretty good assumption. However, if the 2 mutations you're interested in are located on the same chromosome and they are close enough, then things are a little more complicated, because what you need is a recombination event in between these 2 loci to produce your double mutant. And in general, if the 2 loci are far away from each other, this is exactly as if they were independent on 2 different chromosomes. But the closer they are, the less frequently recombination happens. And in reality, your frequency of double mutant production, your efficiency of double mutant production is lower. It's lower than 25%. And this is called genetic linkage. Of course, we can see this by SGA, and we usually use this as a good quality control on our screens to make sure that everything worked out okay. So here I'm showing you an example where a query strain located on chromosome 8 is crossed to a bunch of other loci that are located nearby on the same chromosome. The gene against itself produces a very small colony. Technically, it should give you 0 double mutants, but that's the residual number of cells you get from any type of cross. And then as you cross this strain to other loci that are further away, you get more and more double mutants. And of course, you can quantify this, and we actually don't even have to invent a new system for quantifying it, because it has been around for actually 100 years this year. It's called genetic distance. And the way it's defined is that two loci segregate independently, so in our case produce 25% of the meiotic progeny will be double mutants if they are 50 centimorgans apart. And centimorgan is a measure of genetic distance. It's 50 and not 25 because what in reality genetic distance measures is the frequency of recombinant progeny. And double mutants represent only half of your recombinant progeny, because from every recombination event you get one double mutant and one reconstituted wild type strain. So in reality, 50 centimorgan correspond to 50 recombinants, 50% recombinants, and more generally, you say that two loci are 1 centimorgan apart if 1% of their meiotic progeny is recombinant. So we can do this on all of our data, and we can measure, estimate recombinant frequency and normalize it such that the gene against itself has 0, and then as you move out, recombinant frequency increases. And then you can eventually transform it into an estimate of genetic distance, which involves taking into account the probability of multiple recombination events. And it's a pretty straightforward approach. For reasons of time, I won't go into detail about that. So the good thing is that every good SGA experiment that you do looks something like this. So when you have physical distance from the query locus on the x-axis, and for example, estimated frequency of recombinant or the final genetic distance on the y-axis. And as you move out from that, as gene pairs become more and more distant, both measures increase, which is exactly what you expect. When you look at it, many of these data sets, and we do it for every single screen that comes through SGA, you notice that the shape and the size of these curves could vary quite a lot. Here I'm showing you two examples of two loci, two query strains located on chromosome 15. And one of the most obvious questions that you want to ask is what determines this difference in shape and size? And the first thing that comes to mind is recombination frequency in that particular genomic locus or in that particular genomic region. And the next obvious question is, can we actually use this data to study patterns of meiotic recombination? And I wouldn't be here asking this question if the answer was no. So we think the answer is yes. And I'm going to show you what kind of information we can get out of this data. So we took about 3,800 SGA screens, half of which were published and half of which were unpublished at that time, and we generated a 5 KB resolution genetic linkage map of the yeast genome. What I mean by genetic linkage map in this case is we have 16 of these maps, one for each chromosome, where you have chromosomal coordinates on the x-axis, chromosomal coordinates on the y-axis, and at each intersection I'm telling you the average genetic distance between loci located in those two positions. And you can see there's a couple of things that become pretty obvious when you look at this. First of all, is that linkage is not constant throughout the chromosome, which is known. Recombination rate varies from place to place. You see a large linkage block where recombination happens very rarely, and they're usually separated by regions where linkage is very weak, and they likely correspond to recombination hotspots. And I'm not going to show you the data, but we mapped pretty much every known hotspot that has been obtained through other means onto these maps and shown that at least 60 or 70% of those map to loci that we would predict to be recombinants, and the rest, it's an open question. We are investigating that, whether we're detecting new hotspots or there is some differences in the experiments, the way we do the experiments. The second obvious thing is that there is always a linkage block around the centromere, which is known. It's a known fact. It's a good thing to recapitulate that. In fact, it is known that recombination is inhibited around the centromere, most likely because of all the different things that happen around the centromere during meiosis. But there's also a few interesting new things that we noticed by looking at this. The first one, it's obvious pretty here, is that the centromere is not centered in the middle of its block. So we don't really know why that happens, but it kind of suggests that the centromere positioning is not what drives the formation of the linkage block. Second observation is that the linkage block, like in this case, is usually the biggest block on the chromosome. So it's the region where recombination inhibited the most, and that was never been observed before. The third new observation is becomes apparent when you compare different chromosomes to each other. So here I'm showing you chromosome 6, one of the smallest, and chromosome 15, I think, one of the largest. And the two maps are drawn to scale. So because chromosome 15 is so much larger, I'm only showing you a small portion of it, the portion that's centered on the centromere. As you can see, the linkage block around the centromere on chromosome 15 is much larger than the chromosome 6. And one question that we ask is, well, this is really, really intriguing. Does that hold true for all the other chromosomes? So we decided we're going to quantitatively measure this. And we're going to do this by looking at all the pairs that are located on opposite arms of the centromere. We call it trans pairs, and we're only going to look at the ones that are genetically unlinked. And we're not going to ask what their physical distance on average. And we're going to compare this to pairs that are located on the same arm for control using the same strategy. They're genetically unlinked. We're going to take all these pairs and measure their average physical distance. When we compare the two and we plot them against chromosome size, we see that the trans pair's physical distance is very well linearly correlated, and it holds very well against removal of any outliers, whereas the cis pairs show this characteristic curve, where it's basically composed of two groups. The majority of them are constant, show no relationship to chromosome size, whereas the four smallest chromosomes show a little increased recombination. And this is known, again, because of time I can't go into detail, but this is exactly what you expect. The cool thing is that we want to make sure this is not an artifact of our data. We went back to take genetic distances from SGD, which is based on 40 years of tetradissection, and exactly the same trend holds true. Same thing for other methods for mapping recombinations, such as double-strand break locations, and everything holds true. So what does it mean, and why is this important? The basic question is. The short answer is that we don't really know. We asked this question to as many meiosis experts as we could, and everybody thinks it's really interesting, but nobody has any great ideas why this could be happening. It's interesting because we think it's most likely has something to do with chromosome segregation. So it is known that frequency and position of recombination events around the centromere is critical for chromosome segregation, in the sense that recombination happens too far and spindle attaches improperly to the centromere. Recombination happens too close and the sister chromatids separate too early, way before meiosis II, where they're supposed to be. And this is actually at the basis of Down syndrome. The other cool thing is that without the spindle checkpoint, which is the mechanisms to make sure that the spindle attaches properly, it has been shown 10 years ago that larger chromosomes missegregate more often than smaller ones. So the authors of this paper actually speculated at the end that this could be true, this could be caused by the fact that recombination occurs further away from the centromere on larger chromosomes, and the centromere is more mobile in space and has more trouble binding the spindle. We basically confirming their hypothesis. And the third observation is that we know that recombination near centromeres is associated with the persistence of cohesin complexes that are in charge of keeping the sister chromatids together. We went back to see if cohesin tends to form larger clusters on larger chromosome, and the answer is yes. You have a meiotic cohesin that it's clusters of meiotic cohesin linearly correlated with chromosome size, whereas mitotic cohesin doesn't show that. So in summary, I've told you a story that we've recently published a few months ago in G3, that SGA provides not only genetic interactions, but also linkage maps. That these maps show you that linkage around centromeres correlate with chromosome size, and this could be linked to the accuracy of chromosome segregation. And I wanted to highlight that all of this analysis was based on basically free data that we would normally throw away after performing our quality controls, but in this way we're recovering it and making the best use we can. So I wanted to, of course, acknowledge all the people involved in this project, in particular Charlie Boone, my former supervisor, Brenda Anders and Michael Costanzo, who were directly involved in producing the data, my collaborators at the University of Minnesota and University of Sussex, my little team at Princeton University, and the Lewis Sigler Fellowship, for which I would like to advertise there is openings available. If you're not familiar what a fellowship is, it basically gives the opportunity to recent PhD graduates to start their own lab independently with guaranteed funding and with a little teaching involved. And we have positions open. Thank you very much.
Unfortunately, we're not going to have any time for questions because we went way over the time.
Okay.
So.